look and see what these drugs actually do. And that is called their mechanism of action. How do these drugs work? How do they disrupt microorganisms? So that's what we're going to look through several different classes for that. Okay, so mechanisms of action, of antimicrobial action. The goal of these drugs is to disrupt cell processes or structures of the microbe. So if you remember, we focused on, or we highlighted selective toxicity. Overwhelmingly, we are looking for chemicals that are selectively toxic to the pathogen, not harmful to the host. And that is our overwhelming goal, the selective toxicity. So how do we do that? We're going to have to target things of the microbe that is different than the host. Metabolic processes or structures or proteins or DNA or something that's different in the microorganism than the host. Then if we have a chemical that looks for those differences, that only attacks those differences, then that chemical, that drug will be selectively toxic. So now what we're going to do is we're going to go through 5 different categories of mechanisms of actions. We're going to look at how these drugs work against these microorganisms. We can put them into 5 different categories and based on how they work, their mechanisms of action. We have drugs that inhibit the cell wall, the making of the cell wall, so cell wall synthesis. These are bacteria cell walls and how they do that. We don't have a cell wall, right? No peptidoglycan for us. So this is one category. We're going to look at drugs that inhibit protein synthesis, how these microorganisms make their proteins. We're going to look at another category and drugs in this category that disrupt components of the cell membrane. We're going to look at drugs that inhibit metabolic pathways. So we did a whole chapter on metabolism, cellular respiration, but as we're learning in lab, these bacteria have different metabolic pathways. They can use different things. And so we're looking at what they can do. Can they mannitol salt agar? Can they ferment mannitol? We're looking at all these different types of things, enzymes they make, and their metabolic pathways. And if we can target ones that are different, then that may be a good drug. We're also going to look at the category, some drugs in a category that inhibits nucleic acid synthesis. Now, all of these are looking, the paramount important is selective toxicity. We want it to be able to target the pathogen without harming the host. This is a picture of this with all of our different categories, and you can see inhibition of cell wall synthesis. I got a lot of choices. Why do you think that is? Well, the bacteria have a cell wall. We don't. This makes bacteria cell wall a good target for drugs. Things that damage a bacteria cell wall are not going to harm the host. We don't have a cell wall, so it's not going to harm us. Therefore, we have a lot of drugs in these categories. In this category, inhibition of cell wall synthesis. Protein synthesis, we do have some, little less. Cytoplasmic membrane, not much at all. Metabolic pathways, again, we have to target metabolic pathways of the pathogen that are not the same metabolic pathway of the host. Nucleic acid synthesis, and then over here is some recognition. If we can, a lot of our antiviral drugs are in this recognition. If we can keep these viruses from attaching, then no problem for that, okay? All right, so that's what we're headed into. I'm going to stop this video and we'll break them up into smaller videos, and we're gonna come look at each of these categories, and as we go through, you are looking for selective toxicity. Is this good, this category, drugs in these category, is this good in regards of selective toxicity? Is this bad in regards to selective toxicity? Or is it in the middle? If it's good for selective toxicity, the drugs are targeting something the host doesn't have. It makes it great for selective toxicity. If it's poor for selective toxicity, it means that the drug interferes with the pathogen, but it could also harm the host. That makes selective toxicity hard to come by. So we're going to look at each of those categories and evaluate them in regards to selective toxicity. Okay, the first one will be cell walls. I'll see you at that video.